The autonomic nervous system, or ANS, can be defined as a motor nervous system that controls glands, cardiac muscle, and smooth muscle. It's also called the visceral motor system to distinguish it from the somatic motor system, which controls the skeletal muscles. The primary target organs of the ANS are the viscera of the thoracic and abdominopelvic cavities and some structures of the body wall, including cutaneous blood vessels, sweat glands, and piloerector muscles. The ANS usually carries out its actions involuntarily without conscious intent or awareness, in contrast to the voluntary nature of the somatic motor system. The autonomic nervous system is divided into two main parts: the sympathetic nervous division and the parasympathetic nervous division. These divisions differ in anatomy and function, often innervate the same target organs, and may have cooperative or contrasting effects on the organs they innervate. The sympathetic division adapts the body in many ways for physical activity. Its primary role is to prepare the body for rapid and intense physical activity. It increases alertness, heart rate, blood pressure, pulmonary airflow, blood glucose concentration, and blood flow to cardiac and skeletal muscle. At the same time, it reduces blood flow to the skin and digestive tract. These effects prepare the body for fight or flight responses during threatening, stressful, energy-demanding situations when an animal must attack, defend itself, or flee from danger. It ensures that energy is readily available, prioritizes essential functions, and temporarily suppresses non-essential processes. In humans, this reaction occurs in many situations involving arousal, exercise, competition, stress, anger, trauma, or fear. The sympathetic division is also called the thoracolumbar division because it arises from the thoracic and lumbar regions of the spinal cord. The primary neurotransmitters involved are norepinephrine, also known as noradrenaline, and epinephrine, commonly known as adrenaline, which are secreted by the adrenal medulla. The sympathetic nervous system, or SNS, has several key effects on the body. Firstly, it increases heart rate and blood pressure. This occurs because the SNS releases norepinephrine, which binds to beta-1 adrenergic receptors on the heart. This increases the heart rate, also known as chronotropy, and the force of heart contractions, known as inotropy. The result is an increase in cardiac output and blood pressure, enhancing blood flow to muscles and vital organs during stressful situations. The SNS also dilates the pupils, a condition known as mydriasis. Norepinephrine stimulates alpha-1 adrenergic receptors on the radial muscles of the iris, causing them to contract and dilate the pupils. This improves vision in low-light conditions and enhances peripheral vision to detect threats. Additionally, the SNS dilates bronchioles. Norepinephrine activates beta-2 adrenergic receptors in the smooth muscle of the bronchioles, causing them to relax and dilate. This increases airflow to the lungs, improving oxygen intake and carbon dioxide expulsion during stress or physical exertion. Another effect is the inhibition of digestion and salivation. The SNS reduces blood flow to the gastrointestinal tract and decreases the activity of digestive glands through the action of norepinephrine on alpha-2 adrenergic receptors. This conserves energy and redirects blood flow to muscles and the brain, which are more critical during emergencies. The SNS also stimulates glucose release from the liver. Norepinephrine and epinephrine stimulate glycogenolysis, the breakdown of glycogen to glucose, and gluconeogenesis, the production of glucose from non-carbohydrate sources in the liver. This provides a quick source of energy for muscles and the brain during stressful situations. Furthermore, the SNS promotes sweating. Sympathetic nerves release acetylcholine onto sweat glands, particularly eccrine glands, to promote sweating. This helps regulate body temperature during stress or physical exertion through evaporative cooling. Another interesting effect is piloerection, commonly known as goosebumps. Activation of alpha-1 adrenergic receptors on the erector pili, muscles at the base of hair follicles, causes these muscles to contract, making the hairs stand up. Historically, this was believed to make an animal appear larger and more threatening. In humans, it may be a vestigial response and also helps with thermal insulation. Lastly, the SNS relaxes the bladder and contracts sphincters. Norepinephrine acts on beta-3 adrenergic receptors to relax the detrusor muscle of the bladder and on alpha-1 adrenergic receptors to contract the internal urethral sphincter. This prevents urination during stressful situations by inhibiting bladder contractions and closing the urinary sphincter. The parasympathetic division, by comparison, has a calming effect on many body functions. It is associated with reduced energy expenditure and normal bodily maintenance, including such functions as digestion and waste elimination. This can be thought of as the resting and digesting state. Its primary role is to therefore promote rest and digest activities that conserve energy and restore the body to a state of calm. Its effects are designed to support bodily functions that are essential for recovery, maintenance, and long-term well-being, ensuring that the body can efficiently manage and utilize its resources during periods of rest. The parasympathetic division is also known as the craniosacral division of the autonomic system. This is because its preganglionic fibers originate in the brain, specifically in the midbrain, pons, and medulla oblongata, and in the 2nd through 4th sacral levels of the spinal column. The primary neurotransmitter for the PNS is acetylcholine. Acetylcholine binds to acetylcholine receptors, which are subdivided into 2 types: metabotropic muscarinic receptors, which are G-protein coupled receptors, and ionotropic nicotinic receptors, which are ion channels for sodium and calcium. Muscarinic receptors are G-coupled protein receptors involved in the parasympathetic nervous system. The only exception to these receptors is the sweat glands, which possess muscarinic receptors but are part of the sympathetic nervous system. Nicotinic acetylcholine receptors, or NHHRs, are receptor polypeptides that respond to the neurotransmitter acetylcholine. Nicotinic receptors also respond to drugs such as the agonist nicotine. They are found in the central and peripheral nervous system, muscle, and many other tissues of many organisms. The parasympathetic nervous system, or PNS, has several significant effects on the body. Firstly, it decreases heart rate and blood pressure. The PNS releases acetylcholine, which binds to muscarinic receptors, primarily M2, on the heart. This slows the heart rate, known as negative chronotropy, and decreases the force of heart contractions, known as negative inotropy. This promotes relaxation and energy conservation by reducing cardiac output when the body is at rest. The PNS also constricts pupils, a condition known as miosis. Acetylcholine acts on muscarinic receptors, specifically M3, in the circular muscles of the iris, causing them to contract and constrict the pupils. This protects the retina from excessive light and enhances near vision for activities such as reading. Additionally, the PNS constricts bronchioles. Acetylcholine binds to M3 muscarinic receptors in the smooth muscle of the bronchioles, causing them to constrict. This reduces airflow to the lungs when high levels of oxygen are not needed, conserving energy. Another effect is the stimulation of digestion and salivation. Acetylcholine stimulates muscarinic receptors, specifically M3, on salivary glands and throughout the gastrointestinal tract. This increases the secretion of saliva and digestive enzymes, as well as enhances gastrointestinal motility. Parasympathetic action helps in digestion and absorption of food by increasing the activity of the intestinal musculature, increasing gastric secretion and relaxing the pyloric sphincter. It is called the rest and digest division of the autonomic nervous system. This facilitates the breakdown and absorption of food, promoting nutrient uptake and overall digestion. The PNS also promotes glycogen synthesis in the liver. Acetylcholine and other signaling molecules promote glycogenesis, the formation of glycogen from glucose in the liver. This stores excess glucose as glycogen for future energy needs, ensuring a stable energy reserve, while the primary control of sweating is through the sympathetic nervous system. The parasympathetic nervous system indirectly reduces sweating by promoting a state of relaxation and reducing overall stress. This maintains homeostasis and prevents unnecessary loss of fluids and electrolytes. Lastly, the PNS contracts the bladder and relaxes sphincters. Acetylcholine acts on M3 muscarinic receptors in the detrusor muscle of the bladder, causing it to contract. It also relaxes the internal urethral sphincter through M2 and M3 receptors. This facilitates urination, promoting the excretion of waste products and maintaining fluid balance. The sympathetic and parasympathetic divisions typically function in opposition to each other. The sympathetic division typically functions in actions requiring quick responses, while the parasympathetic division functions with actions that do not require immediate reaction. The functions promoted by activity in the parasympathetic nervous system are associated with our day-to-day living. The parasympathetic nervous system promotes digestion and the synthesis of glycogen and allows for normal function and behavior. Parasympathetic action helps in digestion and absorption of food by increasing the activity of the intestinal musculature, increasing gastric secretion, and relaxing the pyloric sphincter. It is called the rest and digest division of the autonomic nervous system.